Hello, this is lecture number 14, protein targeting to the ER membrane. You'll recall in lecture number 13 we talked about protein targeting to the ER lumen. Originally I had this lecture as part of that lecture. It was all part of 13, but I really wanted to separate this out so that in this lecture we can really kind of start again and focus on protein targeting to the ER membrane. So this is a fairly short lecture, but it's a really important lecture, and frankly I think it covers one of the more conceptually difficult things that we talk about this semester. So as this introductory slide here shows, a lot of proteins are transmembrane proteins, and so we've got to figure out how those proteins get inserted into the membrane. Now, before we do that, there's another really important concept here. So let's spend a little bit of time looking at this because you can't miss the significance of this, or what we're about to talk about won't make any sense. So here's the deal. Look at a protein that's going to go to the cell exterior. So down here. So this is outside the cell. We could actually put plasma membrane over here. And I would say if we actually look at this block here as the plasma membrane, then the protein could be inserted into the plasma membrane, or it could go outside the cell. Now, if a protein is going to go to the plasma membrane, look at its route to the plasma membrane. It's going to come to the plasma membrane from the Golgi through a vesicle. So vesicles are going to emanate from the Golgi and transport that protein over to the plasma membrane. If a protein is going to the lysosome, notice that it's also coming from the Golgi apparatus. Now, this is a double-headed arrow, so some proteins can come from outside, but we're not talking about those that come from the outside of the cell. We'll get to that later this semester. We're talking about those that are coming from the inside. And so in this case, the protein is going to move this way to a late endosome and then over to a lysosome. So the point here is this: if a protein is going to end up either in the plasma membrane, let's say right here, or outside the cell, or over here in the lysosome, it first has to go through the Golgi apparatus. And this is really important. There's no alternative. There's no mechanism for getting proteins directly to the plasma membrane by bypassing the Golgi. And there's no way to get it to the lysosome by bypassing the Golgi either. But so if you go up here and you look at the Golgi and you say, okay, these proteins came from the Golgi, we'll look at the Golgi. The Golgi is going to get its proteins from the ER. So again, you can see that proteins that can go from the ER to the Golgi, and then some of those proteins can go back to the ER. We'll see how that works. But really what we're focused on here is proteins that are going to end up in these excuse me, in these other compartments. This whole pathway right here is called the secretory pathway. Because any protein that's going to be secreted from the cell is going to follow this particular route. In other words, from the ER to the Golgi to the plasma membrane. Now, we're going to spend a lot of time later this semester, actually in the not too distant future, talking about the secretory pathway. But the reason I bring it up here is because if a protein is going to go to the Golgi or lysosomes or the plasma membrane or outside the cell, it first has to go to the ER. So if it's a luminal protein, it's going to go to the lumen of the ER, and then it can move to the Golgi and eventually outside the cell. We'll get to that a little bit later on. But now what we're talking about is plasma membrane. We're talking about membrane proteins. So membrane proteins are not going to be secreted because they're always going to be attached to the membrane. But a lot of those proteins are actually going to function excuse me, at the plasma membrane. So the way the protein gets incorporated into the plasma membrane or into the membrane is very important. Once it gets incorporated into the membrane, it cannot change its orientation. Its original or initial incorporation into the membrane is going to take place here at the ER. So the point is simply this. You're going to have membrane proteins that are going to work in the lysosome, in the Golgi, and at the plasma membrane. Those proteins are going to be inserted into the membrane at the endoplasmic reticulum. And once they're inserted into the membrane at the endoplasmic reticulum, their orientation cannot change. So we've got to spend some time talking about how proteins get inserted into the ER membrane. Again, not just because these proteins are going to function in the ER. I didn't say that. Some of these proteins are going to function in the ER, but some of them are going to end up leaving the ER and go to other places in the cell. But the way they get incorporated into the ER membrane is going to determine how that protein lives out its life. So what are these proteins that we're talking about? Well, some of them are going to be enzymes. We can see that all the way over here to the right. Some of them are going to be receptors that are going to function at the surface of the cell to bring things inside, or they may function at the surface of the cell as part of a signaling pathway. Some of them are going to be anchors that just hold things in place. Some of them are going to be transporters. Now, we'll talk about, especially about transporters and receptors a little bit later on. But the proteins that we're talking about are going to function in a variety of ways. They're going to do a lot of different things. What these proteins all have in common is that they're transmembrane proteins. Trans meaning across, so they're going to span the membrane. And again, not at the risk of making a point that I've already made, let me just say that no matter where that protein functions, whether it's the ER, the Golgi, the lysosome, or the plasma membrane, the way it gets oriented into the membrane is going to be determined when the protein goes to the ER. So let's take a look at some examples of this to illustrate how big a deal this is. Let's look at these receptors over here. Here are 4 receptors, or 3 receptors in another protein, but let's focus on the receptors. Let's take one that we all know about, the insulin receptor. The insulin receptor is obviously going to bind insulin. That insulin is going to play a big role in glucose metabolism in the cell. So that insulin receptor has to be inserted into the plasma membrane in the right way. So imagine that this is a cell. Here's the nucleus. We'll draw the insulin receptor like this. We'll draw a little cup there, and that will serve as the binding site for insulin. So we'll say this is insulin right here. Okay. Now, in order for that protein to function like that, this binding site for insulin has to be outside the cell. If this insulin receptor was oriented like this, it obviously wouldn't work because the binding site for insulin would be on the wrong side of the membrane. So you've got to get that protein oriented into the membrane in the right way. And again, this event of getting the protein oriented in the membrane in the right way is going to take place in the ER. The orientation of the protein in the membrane is referred to as its topology. And so there are several different topological classes of transmembrane proteins. So let's look at these classes. The insulin receptor that we just talked about is an example of a type 1 transmembrane protein. So what is a trans 1 membrane protein? Well, when that protein is inserted into the ER, by the way, this is the ER membrane right here. Okay, so this would be the lumen, and this would be the cytosol. So a transmembrane protein, a type 1 transmembrane protein, is going to have one transmembrane domain. It spans the membrane one time. The amino terminus of that protein is in the lumen of the ER, and the carboxy terminus is in the cytosol. Now that's true for the insulin receptor, the growth hormone receptor, and the LDL receptor. Now let's take a look at the transferrin receptor. This is an example of a type 2. We already know exactly what the transferrin receptor does. The transferrin receptor is also what's called a single pass transmembrane protein, only has one transmembrane domain, but notice that its orientation or its topology is the opposite of a type 1. In this case, the amino terminus is in the cytosol and the carboxy terminus is in the ER lumen. So several proteins are going to end up being type 2 transmembrane proteins. Now again, this is insertion into at the ER membrane. Now let's look at type 3. Now you might, if you look at type 1 and you look at type 2, you might say, well, those are the only two options. Your amino terminus can only be in the lumen or the cytosol. Well, if you look at type 3, you're going to see that the lumen is in the the amino terminus is in the lumen and the carboxy terminus is in the cytosol. So its topology or orientation in the membrane is identical to a type 1, but the mechanism for getting it inserted into the membrane is different. Another big difference, which is not shown very well on this slide, is that in the case of a type 3 transmembrane protein, the domain that's over here inside the membrane tends to be relatively short. Now this doesn't depict that very well. In fact, if I was going to draw this, I would draw it like this. There's our transmembrane like this. In other words, we'll make this the transmembrane domain right here. Here's our carboxy terminus. Here's our amino terminus. But notice when I've drawn it with that amino terminus very short. It doesn't change the topology. The topology is the same. It's just that amino terminus tends to be very short. Now the other option is that it's what's called a multipass. This is a multipass transmembrane protein. In other words, it has several transmembrane domains, several domains that span the membrane. So this protein is like it's stitched into the membrane. And this is called a type 4. Now we'll subdivide the type 4 into a 4A and a 4B, but we'll wait to do that a little bit later on. Okay? A signal anchor is the transmembrane segment of a protein that anchors it to the membrane. So in other words, all these segments right here that span the membrane are called signal anchors. All of these, because they're anchoring the protein in the membrane. So it's a transmembrane domain that spans the membrane. Okay? Now, the topology and the naming of these transmembrane domains is based on the way the protein is oriented in the ER. But as we're going to see on the next slide, this can get a little bit confusing because the way it's oriented in the ER is going to be not necessarily what you might think when you consider the way it's oriented at the plasma membrane. So let's just remember that the insulin receptor, let's use it as an example again, and remember it's type 1 with the amino terminus in the lumen. Okay? So let's go ahead and put up here our insulin receptor. Is a type 1. That means the amino terminus is in the lumen of the ER, and the carboxy terminus, we'll do it like that. Now if I'm going to do that, I need to do this like this, right? Is going to be in the cytosol. All right. Now there's a really important concept here called the conservation of membrane faces. At the outset, this is going to look like it has nothing to do with membrane topology, but trust me, it has everything to do with membrane topology. So let's go over here and look at the ER. So we're at the ER right here. Okay. We know that every membrane, you know this from basic biology, is a bilayer, right? It has there are two leaflets to that membrane. We're not saying it's a double membrane. We're saying that each membrane has two leaflets. It's a phospholipid bilayer. These represent phospholipids. Like that. So we've got two leaflets to that membrane. So what's shown here is the ER with its two membrane leaflets. Now, our tendency when you have an organelle like this is to call this the outer leaflet, and we'll use yellow here, and to call this, the kind of pinkish there, the inner leaflet, because it's on the inside next to the lumen. That really does not work. Now, you may say, well, it looks to me like it works because the outer leaflet is on the outside and the inner leaflet is on the inner side. But trust me, we've got to rename them. And the way we're going to rename them is we're going to call the leaflet that touches the cytosol the cytosolic leaflet. And the leaflet that touches the lumen, we're going to call it the exoplasmic leaflet. Okay? Now, before we go on this slide, let me show you why you've got to do that. Let's take the mitochondria, for example. The mitochondria is a double membraned organelle. So there's the outer mitochondrial membrane. The outer mitochondrial membrane has two leaflets. So let's go ahead and draw the two leaflets. Now, if it was that simple, we could call this the outer leaflet, and we could call this one the inner leaflet. Okay? But we said that it wasn't that simple, and you'll see why here in just a second. So let's get rid of that because we're not going to call them like that. Now remember, the mitochondria is a double membrane, so it has an inner membrane with those cristae, right? That membrane also has two leaflets. So let's draw the two leaflets in here. Okay, this will illustrate why we've got to change the nomenclature. Now, if we call this the outer leaflet and this the inner leaflet, the question is, what would we call the two leaflets of the inner mitochondrial membrane? Well, you could call them the inner and the outer leaflet of the inner mitochondrial membrane. But you'll see right now why that's not going to work. So let's use the correct nomenclature. So we're going to call this the cytosolic leaflet. Okay. And we said that this then is going to be called the exoplasmic leaflet. Now, two membranes or two leaflets of a membrane that face the same compartment have to have the same name. So if we look at the outer leaflet of the inner mitochondrial membrane, what are we going to call it? Well, it faces the same compartment as the inner leaflet of the outer mitochondrial membrane, therefore it has to be called exoplasmic. So that's the exoplasmic leaflet of the inner mitochondrial membrane. Now, in terms of naming, we only have two options, exoplasmic and cytosolic. So what does that mean that the inner leaflet of the inner mitochondrial membrane is called? It's called the cytosolic leaflet as well. Now that's a little bit counterintuitive because you would say that leaflet of that membrane is about as far from the cytosol as you can get, and you would be correct. But that's the nomenclature that we use, and that nomenclature is going to make a little more sense here in just a second. Okay, so you got the nomenclature down. So now let's go back. I'm going to erase that. Okay. Let's also erase this, get this out of the way. And look at what's going to happen. Now this again is the ER. We've named those leaflets of the membrane. Now imagine that we're going to take a protein and we're going to move that protein over to the plasma membrane. Remember, this is going to be part of the secretory pathway. We're going to incorporate those proteins into vesicles. Any protein movement beyond the ER is going to be vesicle mediated and is going to go through these vesicles that are part of the secretory pathway. So let's just assume we've got a protein not in the lumen of the ER, but inserted into the membrane. Now let's use our insulin receptor as an example. We said it's a type 1 protein with the amino terminus in the ER lumen. So let's put the amino terminus, let's use yellow here, like we did on the previous slide. So we'll put the amino terminus, whoops, let's do it over here. We'll put the amino terminus in the ER lumen. You know what, I'm going to go back and change the color to make it easier to see. Let's try this dark blue. Yeah, that's better. So we'll put the amino terminus in the lumen, the carboxy terminus here. Now when a vesicle forms, we're going to conserve those, we're going to conserve the membrane faces. Now what that means is that a leaflet that is a cytosolic leaflet will forever be a cytosolic leaflet. And a leaflet of the membrane that is an exoplasmic leaflet will forever be an exoplasmic leaflet. So that means when this vesicle forms, the cytosolic leaflet, which is the darker red, is going to be on the outside. It's still facing the cytosol. The exoplasmic leaflet faces the lumen. So that makes perfect sense. Because remember, these are named based on the compartment that they face. It's easiest to remember with a cytosolic leaflet. Now let's take our insulin receptor and assume that it gets incorporated into this vesicle. So here it is. Remember, once the orientation is established at the ER, it never changes. So the orientation hasn't changed. The amino terminus is still in the lumen. Now if that protein were to be incorporated into that vesicle, it's going to be incorporated like that. In other words, the amino terminus is still in the lumen. Now that vesicle is going to fuse with the plasma membrane. If it fuses with the plasma membrane, it can fuse in one of two ways. It can either fuse and open up so that the contents, the luminal contents, are exposed to the outside of the cell, or it could fuse and open up with the luminal contents exposed to the inside of the cell. Now, the purpose of these vesicles, these secretory vesicles, is to dump things to the outside. So if it's going to fuse with the membrane and open up with the contents toward the inside, you can't accomplish that goal. That would just release the protein back out into the cytosol. So rather, what's going to happen is it's going to fuse with the plasma membrane and open up in such a way that it dumps its contents outside the cell. Now that also conserves membrane faces. So imagine this vesicle coming along right here. It's going to fuse with the membrane, and the cytosolic leaflet of the membrane is over here, and the exoplasmic is over here. Okay, now that means, we'll talk about this more in class because I've got this illustration where I use my hands and I think it is really helpful, but I can't do that right here. But remember, the orientation of this protein is really is important and can't change. So that means that it's going to be oriented like this. Now you see why this is a little bit counterintuitive, because what this means is that the amino terminus started out over here in the ER, in the lumen of the ER, but it's going to end up on the outside of the cell. But that is consistent with everything we know about protein insertion into the membrane and the way they work. So let's again think about the insulin receptor. It's a type 1. Its amino terminus is in the lumen of the ER. So here's the question that I would ask you. The portion of the insulin receptor that binds insulin, is that going to be on the amino terminal side of the membrane or the carboxy terminal side? It's got to be on the amino terminal side, right? So let's put that kind of cup on this side, which we said is going to serve as the insulin binding site. There it is. Now here comes insulin from outside the cell, and the insulin can bind right there. Now you can also see why this is so important, because what if you inserted that insulin receptor into the membrane in the wrong orientation? So let's use light blue here. Let's say that it was put in the wrong way. And so if it's now incorporated into a vesicle, it's going to look like this. And when it gets over here to the membrane, what's its orientation going to be? Now that's a problem, because we said that the portion that binds the insulin is on this side, on the amino terminal side. So it's not going to work. So you can see how important it is for proteins to get inserted into the membrane with the right orientation. This concept of conservation of membrane faces will come up over and over and over again. So now what we need to do, now that we understand how proteins are inserted into the membrane, or why insertion with the right topology is important, is let's go back and look at the mechanism. Let's see how this works. So we're going to look at all of these different types and see how it works. We'll start out with type 1, then we'll do 2 and 3, and then we'll do type 4. Now, it turns out that type 1 is pretty easy. Type 1 is going to have a signal sequence on the amino terminus, just like it was going to the ER lumen. So it's a typical signal sequence. Now in this diagram, notice that it's called a start transfer sequence. It's called a start transfer sequence because the sequence that comes after it goes through the translocon into the ER lumen. So it's a typical signal sequence, but when it comes to putting transmembrane proteins in the membrane, it's going to be referred to as a start transfer sequence. So this protein is going to move through the translocon using the mechanisms that we described before. There's nothing new here. We're going to use SRP, the SRP receptor, just like we did before. We're going to use the energy of translation. We're going to use signal peptidase here to cleave off the signal peptide. And then the protein, the energy of translation, is going to force that protein through the translocon. Here's the only difference. There's a second sequence. It's an internal sequence, and this internal sequence is called a stop transfer sequence. It's called a stop transfer sequence because when that sequence gets into the lumen, gets into the translocon, the translocation stops. Now, why would the translocation stop? Well, remember, the translocon actually has an opening on the side, a seam that will open where things can migrate out. So that opening is exposed to the membrane. Well, these lipids in the membrane are very nonpolar. So it turns out that this sequence right here, this stop transfer sequence, is a nonpolar sequence that's about 25 amino acids long. Now, why does it matter that it's 25 amino acids? It matters because 25 amino acids is about how many amino acids you need to create a segment that's roughly equivalent to the width of the membrane. Okay, so in other words, this is the width of the membrane, and it would take about 25 amino acids to span that width. So again, to get a type 1 transmembrane protein inserted into the ER membrane is fairly straightforward and fairly simple. You're going to use the same mechanism that you use to get a protein into the ER lumen. The only difference is you're going to have an internal stop transfer sequence that's going to stop the transfer. Now, as the ribosome continues to translate that mRNA, it continues to make the protein, but everything that comes after that stop transfer sequence is going to end up out here in the cytosol. Okay, so this is a type 1. Again, that's exactly what we saw for the insulin receptor. The LDL receptor is another example with the amino terminus over here in the ER lumen, which, when it gets to the plasma membrane, is going to end up being outside the cell. Now, let's look at a type 2 and a type 3. We're going to kind of look at these together. So kind of put type 1 off to the side because we know how that works. It's based on the mechanism for getting things to the lumen. Now, type 2 and type 3 have an opposite orientation. Remember, with type 2, the amino terminus is in the cytosol, and with type 3, the amino terminus is in the lumen. So now we're going to see how they get inserted into the membrane. So let's start out with a protein. In this case, the mRNA is being translated, but there is no signal sequence on the amino terminus. So as far as that ribosome is concerned, it's translating an mRNA that's encoding a protein that's going to be dumped into the cytosol at this point, because there's no amino terminal signal sequence. But as it continues to translate that, there is an internal signal sequence. Okay, this internal signal sequence is going to be recognized by SRP. And so everything that we've talked about is going to happen. We're going to pause translation, and now we're going to move this whole complex over to the ER membrane. We're going to hand that ribosome off to a translocon, just like we would do for luminal proteins. But once that protein is handed off to the translocon, there's a decision that has to be made. And the decision that has to be made is how it gets oriented. So let's look at a type 2. In the case of a type 2, it's going to be oriented with the amino terminus. In other words, this portion of the protein that's already been made is going to be on the cytosolic side of the membrane. Now, how does it make that decision? Well, it turns out that there are positive charges. It's hard to see that yellow. Let's use this purple, see if it works a little better. Yeah, there's a positive charge right there. Positively charged amino acids. The positive charges are always going to be on the cytosolic side of the membrane. So always on cytosolic side of the membrane. Okay, now at this point, the ribosome is sitting on top of that translocon. SRP is gone now, so translation will resume. And now we can use the energy of translation to move the rest of it, this portion, as it's being made, on through the translocon into the lumen. And so the end result is going to be a typical type 2 transmembrane protein with the amino terminus in the cytosol, the carboxy terminus in the lumen. Now, let's suppose that when the ribosome takes this protein right here, I should say that a different way. When SRP takes the ribosome and the mRNA and the protein that's already been made over to the ER membrane where it docks with the SRP receptor, and the ribosome is handed off to the translocon, let's assume now that the positive charges are not on the amino terminal side, but they're on the carboxy terminal side. of that internal signal sequence. Remember, the positive charges are always on the outside. So what's going to happen now? Well, in this case, if the positive charges are on the outside, then what's going to happen is, let's put our positive charges right here. That's the way it's going to be oriented. Now, the internal signal sequence, if the positive charges are on the carboxy terminal, are acting like a stop transfer sequence. Because what comes after that is going to be in the cytosol. In other words, we're going to stop transfer through the translocon. This is exactly what we saw with a stop transfer sequence on a type 1. In other words, anytime you have a stop transfer sequence, what comes after that sequence is going to be in the cytosol. So this sequence, everything after this then is going to be in the cytosol. So this is all in the cytosol. Now you might say, well, how does that amino terminus get into the lumen of the ER? It can't get in by the energy of translation because that portion of the protein was already made before it even got over there. This is why it's significant to note that this region that gets inserted into the lumen of the ER is usually short. If that was a really long segment, it's hard to imagine how you could get a long segment into the lumen of the ER. So every time I get to this point, I like to think that, and this is kind of a silly way to look at it, but it helps me kind of think about what's going on. It's as if you take your finger and you put your finger on the end of this protein right here and you just push it through. Because we don't know exactly how that short segment gets in. Somehow or another it gets in. It's probably very significant that it's short because, as I said, if it was long, it would be difficult to imagine how you could get it through. But it gets through with the amino terminus now in the lumen. So the key to a type 2 and a type 3 is this. You're going to have an internal signal sequence that is going to be flanked by positive charges. The positive charges are always going to be on the cytosolic side of the membrane. The other thing before we leave this is to note that that internal signal sequence, if it's a type 2 transmembrane protein, is functioning as a start transfer sequence. If it's a type 3 protein, that internal signal sequence is functioning as a stop transfer sequence. In both cases, no matter whether that internal sequence functions as a start or a stop transfer sequence, it is also functioning eventually as a signal anchor because it's anchoring the protein into the membrane. Now that's type 1, 2, and 3. Let's look at type 4. Now type 4 is a multi-pass. Whoops. Multi, of course, just meaning many. So there are many segments that are going to pass through the membrane. So it's a multi-pass transmembrane protein. There are a lot of these. The G protein coupled receptors we'll get to a little bit later on. We'll talk about those in great detail. We'll talk about glucose transporters. We've already talked about Sec61. Remember the Sec proteins. Sec61 is the translocon. So these are multi-pass transmembrane proteins. So how are they going to get inserted into the membrane? Now, we're going to use this example right here, and this example is about as simple as you can get. This is a multi-pass transmembrane protein, but really it only has 2 transmembrane domains, so it's a double pass. But it will allow us to see what's going to happen. Now, if it's a double pass transmembrane protein, we need to have 2 signal anchors or 2 transmembrane domains. So let's look at how this is going to work. In this case, we're going to have, again, some internal signal sequences. The first internal signal sequence is going to function as a start transfer sequence. The second one is going to function as a stop transfer sequence. So again, everything that we know is going to take place. The ribosome is translating the mRNA. SRP grabs a hold of the binding pocket on SRP, is going to bind to the signal sequence, the internal signal sequence in this case. The translational pause domain is going to do its job. We're going to carry this whole thing all the way over to the ER membrane, where the ribosome will be docked on top of the translocon, as we've seen multiple times now. But what's going to happen is this first sequence is a start transfer sequence. That start transfer sequence, I don't know why they don't show this on this slide, but it's going to be flanked by positive charges right here. Remember, the positive charges are always on the cytosolic side of the membrane. And so that's the way it's going to be oriented into the membrane. Now that's a start transfer sequence. So everything that comes after it is going to move through the translocon. If that's all there was to it, then what would we have here? We would have a type 2 transmembrane protein. That's exactly what we saw on the last slide. So this is exactly like a type 2 up to this point, but now it changes because another internal sequence is synthesized. In this case, it's a stop transfer sequence. And so as that stop transfer sequence gets into the lumen, gets into the translocon, we're going to stop the transfer, which means that everything that comes after it is going to be in the cytosol. So everything that now comes after that stop transfer sequence is out in the cytosol. So what do you have then in the lumen? Well, you have a loop, the loop between your start and your stop transfer sequences. So that's a double pass transmembrane protein. Now, when it comes to multipass transmembrane proteins, we've ignored something up to this point. We haven't said anything about where the amino terminus or the carboxy terminus is. Well, it turns out that for multipass transmembrane proteins, you have to divide them into type 4A and 4B. A 4A protein always has the amino terminus in the cytosol, and this is a 4A. A 4B always has the amino terminus in the lumen of the ER. And you might say, okay, well, what about the carboxy terminus? As far as naming these proteins, the carboxy terminus doesn't matter. You're going to name them based on the location of the amino terminus. So a 4A could have its carboxy terminus in the lumen or in the cytosol, but its amino terminus, by definition, has to be in the cytosol. Now, before we look mechanistically at how this works, let's go down to the bottom, because there's something kind of clever to think about here. If you have an even number of transmembrane domains, the amino terminus and the carboxy terminus will always be on the same side of the membrane. It doesn't tell you which side of the membrane. They could both be on the luminal side, or they could both be on the cytosolic side. So it doesn't tell you whether it's a 4A or a 4B. It just tells you that they're on the same side. If you have an odd number, then your amino terminus and your carboxy terminus are going to be on opposite sides. Once again, it doesn't tell you whether it's a 4A or a 4B. It just tells you they're on opposite sides of the membrane. So that's going to be important when we kind of look at a protein and see how it might get inserted, its orientation in the membrane. Now, what if we have a multipass transmembrane protein that has several transmembrane domains, more than 2? We saw how it worked last time, but now let's look at it for a situation in which you have more than 2. So let's look at the case for a 4A. Remember, a 4A is going to have its amino terminus in the cytosol. So what's going to happen then? Well, we're going to start translating this mRNA. We're going to encounter this first internal sequence right here, this first internal sequence. That internal sequence is going to be a start transfer sequence. The positive charges are going to be on the amino terminal side, which is going to force that amino terminus to be in the cytosol. It's a start transfer sequence, meaning that everything that comes after it is going to be in the lumen. So if we were drawing this protein, okay, let's draw it as it's being made. Here's the amino terminus. Let's do this. Let's label this as the cytosolic side. This is the luminal side. Okay. That's the membrane. Now let's take our protein. The amino terminus is going to be up here. Here's our transmembrane domain. Then what comes after that is going to be in the lumen. Okay, here's our amino terminus. Now this is, you got to kind of use your imagination here to envision what's going to happen. As that ribosome is translating that mRNA, it's now going to synthesize rather another signal anchor. The next signal anchor is going to be a stop transfer sequence. Now remember, what comes after a stop transfer sequence must be in the cytosol. So when that transmembrane domain is incorporated into the membrane, what comes after it is going to be in the cytosol. But again, the ribosome continues to translate it, and now it translates another start transfer sequence. Positive charges here, positive charges here. It's a transmembrane domain. It's a start transfer sequence, so what comes after it is in the cytosol. The next one is a stop transfer sequence. So what comes after that is out here. So we've got a transmembrane domain here, a transmembrane protein rather, with 4 transmembrane domains, which means our amino terminus and our carboxy terminus are on the same side of the membrane. The amino terminus is on the cytosolic side, so this is a 4A. Okay? Now let's go with a 4B. I'm not going to go through the whole thing because it's going to work exactly the same way with one major difference. The first sequence that's generated well, let me back up and go back to 4A for just a minute. Notice with 4A that we have alternating start, stop. You know what? Let's do this. This is start, stop, start, stop. Okay? For a 4A, you just want to kind of alternate that. Now it gets confusing here at the beginning because unfortunately the way this textbook does this is it calls this first transmembrane domain a start transfer sequence. It calls it a start transfer sequence because it's flanked by these positive charges. The positive charges tell it how to get oriented into the membrane. So this is going to be starting out like a type 3 transmembrane domain, type 3 transmembrane protein. But the positive charges are going to put, these positive charges are going to put this transmembrane domain in the membrane. These positive charges are going to put this transmembrane domain in the membrane with what comes after it out in the cytosol. So the way I like to think about this is that if the amino acid sequence that comes after the domain is in the cytosol, that's really a stop transfer sequence. So here we've got a stop, and then a start, and then a stop, and then a start, then a stop, then a start, and then a stop. Now I would encourage you, we'll work on this in class, but I would encourage you to take a piece of paper and kind of draw this out to see how this works. Notice in this case too, we have 1, 2, 3, 4, 5, 6, 7 transmembrane domains. And notice that that's putting the amino terminus in the lumen, making it a type 4b, but we have an odd number of transmembrane domains. Therefore, the carboxy terminus is in the cytosol. Okay, I know that can be a little challenging. It can exercise your ability to visualize what's going on. We'll talk about this more in class, but that's the basis for forming a type 4 and a type 4a and a type 4b. Now, before we end this, we need to do one other thing. There's another mechanism, totally unrelated to the mechanism that we've just described, that is responsible for putting what are called tail-anchored proteins into the ER membrane. Now, probably we wouldn't even spend any time talking about this, except this is the mechanism that's responsible for inserting SNARE proteins into the membranes. Now, SNARE proteins are going to be really, really important when we get to vesicular trafficking. We're not even going to worry about what they do right now. We're just going to see that these are proteins that are going to be inserted into the ER membrane, but they don't actually pass through the membrane. They're just anchored to the membrane. So let's go over here all the way to the end, and we can see what this is going to look like. It's got a signal anchor, but that signal anchor is on the carboxy terminus. So let's look at the mechanism for how this protein is going to be anchored to the membrane. You wouldn't even really call this a transmembrane protein because it doesn't span the membrane. In other words, you don't have part of the membrane on one side and part on the other. You just have it anchored in there. So here's what's happening. The protein is made. It's completely made. So there's nothing at this point, because it's completely made, that causes it to be translocated or moved over to the ER. But rather, when that carboxy terminal signal anchor is made, it's recognized by this thing called a pre-targeting complex. So this is a protein complex that will recognize that carboxy terminal domain. That pre-targeting complex will bind to another protein complex called GET3. GET3 is an ATPase, so it will bind and hydrolyze ATP. At this point, though, it doesn't hydrolyze the ATP. Rather, what happens is GET3 has a high affinity for some transmembrane proteins on the ER called GET1 and GET2. Here's GET1 and GET2. So GET3 binds to GET1 and GET2. And when it does that, by some unknown mechanism, it hands that protein off to the membrane. Or said another way, it inserts that protein into the membrane with the carboxy terminus inserted into the membrane, which leaves the rest of the protein out here in the cytosol. Now, the fact that the rest of that protein is in the cytosol is going to have everything to do with the way these function. So again, we'll see that a little bit later on. Then what happens is the ATP is going to be hydrolyzed. When the ATP is hydrolyzed and the ADP and the inorganic phosphate are released, the GET3 undergoes a conformational change that causes it to be released from GET1 and GET2. It can go back up here and get another protein and do it all over again. So that's a totally different mechanism for getting tail-anchored proteins into the ER membrane.